right on target. Yeah, so I'm going to do my acknowledgements at the beginning. I'd like to thank Mark and Sandy for working with me on these projects and offering ideas all along the way. Also, I've been doing this project in particular with a high school student, Sanvi Prasad. And so thanks, Heiko, for setting up the high school internship program and making all that possible. So the idea, it's adaptive therapy. We all are well aware of adaptive therapy. I guess I should say the title. So I'm looking at how normal tissue and the tumor phenotype could influence adaptive therapy in an imaging scale model. So the background for this model is that this was a model I built to look at CT scan data and try to model based on the information that can be gathered from those measurements. So effectively tissue density. So I'm looking at a sort of imaging scale model paradigm in 3 dimensions. And the idea is that each spatial location here is at the resolution that you'll see in a CT scan, so like cubic millimeters. And so inside of each of those cubic millimeters, there's these different cell phenotypes, so tumor cells, epithelium, endothelium, and glucose, Darcy-Ventrell equation that the different cells are working with. And the way that I'm seeding the model with the density that's in the CT scan is using the basically the density in the CT. I'm saying that that's effectively a measurement of density, and then I'm seeding the endothelial cell population based on that density measurement. And that's a sort of carrying capacity for the endothelial cells, so that's consistent over time. And the endothelial cells, they're the blood vessels, so they in turn situate the amount of resources that are available to the cells in those locations, which ultimately I get the same density in the tissue that I got from the CT scan by controlling the amount of resources that are available locally. So it's kind of a way to turn that imaging data into a model. So I guess, the cool thing here for me, a lot of the coolness here is that I'm able to represent counts in individual cells. And on the scale of, imaging, we're talking about trillions and trillions of cells. So a sort of way to implement that was to use this population grid approach. And the idea there is that in these cubic millimeter locations, we're putting a sort of bin of cells that are all sort of similar. And so you end up with a count of cells rather than individual agents. And then I use a binomial and multinomial calculators, which run in invariant time regardless of the number of individuals. So I can, say like how many cells are going to die in this time step at this one location, and flip my like 10,000 coins all at once and get that number. And then that becomes an update to the system, and then division likewise. And then for movement, because there's multiple different paths the cells could take, I'm using the multinomial calculator instead. And that just gives me, sort of like rolling a bunch of dice instead, and you get the number that will go into these different directions. And on the right, I was showing an example of the model in 3D. So everything, all of them I'm going to be showing is in 3 dimensions, but I'm going to be showing 2D slices for the rest of this. So this is just an example of what the thing looks like in 3D. So I'm showing the sensitive cells are kind of forming the shell around the resistant cells, because we're assuming the resistant cells are, paying a pretty substantial cost. They're moving and dividing much slower. But eventually, you can't constrain them forever because the drug is ineffective against them, and over time they break out. So this is the interaction diagram for this model. And so like I was saying, it's incorporating all these other aspects as well as sensitive and resistant tumor cells, and now we're introducing a drug as well. And the drug is only impacting the sensitive cells, and the drug is also sort of consumed by the cells, which creates this gradient from the outside of the tumor in. So there's more drug on the outside than on the inside. And so this is the 2-dimensional slice view that I was talking about. So the top left is the drug, which is being pulsed for this adaptive therapy scheme. And you can see that gradient from the outside to the inside because the drug is being consumed by the cells. And then to the right of that, you have resistant cells, which are slowly, breaking out bit by bit. And then the sensitive cells are constraining them, and they're being affected by the drug, and that's what's causing this pulse and brightness. The blue is showing complete absence of cells, and black is like a very small number, and white is maximum capacity for that voxel, which is about like 1 million cells. And then there's the glucose, so there's that resource component to this as well, and endothelium, which you can barely see, but there's and about 30 epithelium to 1 endothelium ratio in the tissue, and then the epithelium, which is getting displaced by the tumor. And so the way I was looking at this was to kind of compare continuous to adaptive therapy across a variety of different conditions. And this is showing sort of one example of what that looks like. So I'm running pairs of adaptive and continuous simulations, and then plotting that as a point in this two-dimensional space. So the x-axis here is showing how long it took for adaptive therapy to progress, and then the y-axis is how long it took for progression under continuous therapy. So, and then the black is that sort of the diagonal line where both progression times happen in the same amount of time. So if you're below that diagonal line to the right of it, below to the right, then adaptive therapy is effectively doing better than continuous. And if you're above, then continuous is doing better than adaptive. And I'm looking also at different densities of the normal tissue. So I'm just isolating here two different densities to look at. And there's sort of the high density case, which is where the normal cells are pretty close to the carrying capacity of the voxel overall. And then the low density case where there's sort of a pretty low baseline amount, which is what you'll see in like lung tissue primarily. And then I'm also looking at two different drug cycling thresholds. So the way I'm doing when to switch adaptive therapy is a bit different, I think, from most of what we've seen. I'm using a volumetric calculation. So basically, I'm just saying if there's one or more tumor cells in a location, then it's considered part of the tumor. And my threshold is saying when the tumor volume shrinks below, say 90%, then I stop the drug and let it regrow, or 50%, so it has to shrink to 50% of its original size and let it regrow. So I'm not looking at the number of cells, I'm looking at the amount of volume that this tumor as a whole takes up. So this is all the cases that I looked at. So on the left, we're looking at the 90% switching threshold. On the right, we're looking at the 50% switching threshold. And then each of these subplots is looking at a different phenotype for the tumor. So the tumor cells can modify the environment by being more glycolytic, so they consume more of this glucose than the normal cells do. And they can also become more angiogenic. So they can actually modify that endothelial carrying capacity that I mentioned earlier. So they can basically alter the angiogenic, or they can alter the vascular environment and basically recruit more glucose to the system. So I'm going to first highlight two particular cases here. This is the top left is kind of the normal-like tumor cells that don't recruit more vasculature and consume the same amount of glucose as the normal cells do. And in particular, I'm looking at the low density case because that's where adaptive therapy is doing the best. And so I'm comparing here the constant therapy on the left, and then the 50-50 switching threshold in the middle, and then the 90% switching threshold on the right. And that's, so blue on the top there is constant therapy, and then the orange is the 50% threshold, and then the green is 90%. So you can see that under these assumptions, the 90% threshold does much better in the sort of normal-like tumor case. Okay, and now I'm going to look at with the glycolytic phenotype. So here we're doing just different gradations of the glycolytic phenotype. And again, I'm looking at low density in this case because the effect of this glycolytic phenotype seems to be most pertinent in that context. So in the normal-like condition, right, adaptive therapy is doing very well, but as you increase the glycolytic phenotype, it seems like the adaptive therapy is doing worse and worse. And so what is the reason that underlies that? You can sort of see it looking here, just if I reset the video here. If you look at the far right, you can see the sensitive cells are at a much lower density per voxel than on the left case. And the reason for that is just that they're consuming more of this resource. So there's not enough available for a large density of sensitive cells. And so that means that that wall that surrounds the resistant cells is much less dense. It's weaker it's not as constraining on the resistant cells as they grow. And what I'm showing here is essentially the outer layer around the outside of the tumor. So if you imagine the tumor is like an onion, we're going like one layer in and looking just at the average cell counts in that domain. And then time is in the x-axis here, and cell count is the y-axis. And so you can see on the left where this glucose phenotype is sort of a middle amount, that there's a lot of sensitive cells that are constraining the resistant cells. That's the orange, and then the resistant cells are the blue. And the epithelium is pretty mitigated because of all those sensitive cells outcompeting them. But then when there's a more glucose consumption, both of the sensitive cells drop way down, and our resistant cells have a much easier time breaking out. Okay, so now I'm looking at the angiogenesis case. So we're increasing angiogenesis now, and we're leaving out glycolysis, or at least we're not modifying that phenotype. So in that case, once again, you can see that adaptive therapy is sort of getting closer and closer to that 50-50 line, so it's not doing quite as well. But the reason here is a bit different. You can see on the right that there's still fewer sensitive cells than on the left. But it's not because of a lack of resources, because they're actually recruiting vasculature, so there's more resources available. But what that leads to is that the drug is also entering through the vasculature. So there's more exposure to drug in this case. And also it creates an environment with more resources around. So the epithelial cells are actually able to take over that space and go to a bit higher density than they would be normally. And what that leads to essentially is, instead of competing between resistant and sensitive cells, we're now competing between resistant and the epithelium. And that's sort of what I'm showing here with this layer view once again. So with a middle amount of angiogenic phenotype, you can see that the sensitive cells are sort of doing their job at producing this wall around the resistant cells, and there's not as many epithelial cells present. But then with a lot of angiogenesis, because of the drug, the sensitive cells are very much tamped down, and the epithelial cells are somewhat replacing them, but still it's a much easier battle for the resistant cells to fight against the epithelial cells than the sensitive cells. And the last case I'm looking at is where we specifically are, we have high angiogenesis and high glycolysis, and looking at the two different drug switching thresholds in that context. So basically under constant therapy, it looks more or less like it has throughout. But when we introduce the switching thresholds, things somewhat reverse. So you can see that now the 50% switch is actually doing better than the 90%. And the reason there is a little bit nuanced. You can kind of see it looking here. If you pay attention to the 50% switching threshold and look at the epithelial cells in particular, that when the drug is on and the sensitive cells are killed off primarily, the epithelial cells are actually using the vasculature that the sensitive cells produced to replace them. And then when the drug goes back on again, when the drug goes back off again, the glycolysis of the sensitive cells forces away those epithelial cells. They're outcompeted because of the lack of resources. And so you get this sort of back and forth where sometimes the epithelial cells are using the vasculature the sensitive cells created to form that wall. Other times the sensitive cells are using the angiogenesis to grow and without the presence of drug, and using the glycolysis to keep the epithelial cells out. And so you can see that you have these pretty substantial oscillations of large densities of these different cell types in the left case, whereas in the right case, things are a bit more normalized, so that you get more of that case where there's competition between the epithelium and sensitive cells, moment to moment. And so you don't really get dominance of one or the other. So there's a bit more room for the resistant cells to make it out. And so looking at all the cases and all together, adaptive therapy is performing about as well as continuous therapy, if not better. So at least under these assumptions, it seems like adaptive therapy is a somewhat safe bet, because you'll at least do as well, if not better. And the tumors that are either or glycolytic or angiogenic tend to make adaptive therapy worse. But you get this odd effect when you have both phenotypes or both traits expressed in a single phenotype, where adaptive therapy is actually leveraging both sensitive and normal cells alternatingly to block the resistant cells. And so in the future, I'd like to look at more clinically relevant ways of measuring this tumor burden. So I basically took the most sort of straightforward assumption, because with this glycolytic and angiogenic phenotype, the densities of these cells can vary pretty widely. So because of that, I was saying, if we have one or more cells, then we'll count it as inside the tumor. That way, even if it's a very glycolytic tumor with very few tumor cells, it'll still sort of be counted as within the tumor burden. But a more clinically relevant way of looking at it could be to look at something that would show up on imaging. So look at the density of the normal cells at some baseline level, and then see if there's more than some deviation above or below that in the tumor, then that would be a way of measuring the tumor burden. And another thing to look at is making the phenotypes and resistance evolvable traits rather than one phenotype or the other that you sort of are stuck with from the beginning at the initiation of the system. So yeah, thank you.
Any questions for Rafael? I think we're all just a bit tired, right? We got to the end of it. Well done. Thanks again,